everyone, welcome to another episode of Inside the Sciences. I am Sheena Burhal and I will be your host. And in this episode, we are going to uncover a hidden physical force inside bioreactors that might just be silently sabotaging your most sensitive therapeutic cells. It's called the shear stress. And more importantly, we are going to explore a breakthrough technology designed to fix this very problem. So how do we grow cells that aren't just living, but also stable, strong, and ready for real-world applications? We are joined today by Fitra Zara Salsabila, a bioprocess engineer here at Esco, to help us answer this problem. Hi, Fitra.
Hi.
Hello, welcome to Inside the Sciences. So we're going to talk about shear stress today, a problem that is, faced by bioreactors in industries, specifically for most sensitive cells. So, but before we go any further, can you tell us what exactly, because it's a bit technical, what exactly do you mean when you say shear stress? Why is it important?
Okay, for the shear stress is a kind of force that happens when liquid like the culture media in bioreactor move and rubs against the surface of the cell. So that rubbing creates pressure which can affect for the cells grow or survive.
Alright, so it's more like a fluid rushing over the cells.
Yeah, it's exactly. But the friction makes seem small, but the fragile cells like stem cell or cells used to make the vaccine, it can be harmful. So as we know about the traditional bioreactor like stir tank or bubbling, there's a lot of stirring or bubbling in the bioreactor. Exactly. So it make them damage or detach if the way to make them as the cells.
Alright, so it sounds like, managing shear stress isn't just about, technicalities. It's more like a key to having healthy cells and cells that could actually do their jobs. Is that right?
That's right. When you're working with delicate cells that need to stick a surface to the cells, they need to really the environment grow cells like for the keep the gentle or anything else that the cells grow properly.
Alright, so we know that shear stress can really be damaging for cells, but what exactly does it do to the cells? What are the real tangible effects?
Okay, for the in small and controlled amounts, also shear stress can be helpful. It's like mix for the nutrients, help for the aeration, so can grow the dam properly. But if the force get too strong, actually it's make there the cell outer membrane, so make the damage for the inner structures like cytoskeleton or in the until the high resources. So because the cells is like the burst, so that we call the lysis. So it's kind of very fine balance for even for a sensitive cell, even moderate the cells can be a big risk to their health and function.
Even moderate shear stress. Okay, so it's not just about the cells dying. It's like the functions are also messed up.
Okay, for even the cells don't die immediately, so it can make the internal function get disrupted. So that can lead to lower productivity, poor quality until the unpredictable results. So which is the last thing you want when making something as important like therapies or vaccine.
Alright, so I also learned that, from the previous episode also about bioreactors, we learned that not all types of cells can handle the shear stress the same way, because we have the suspension cells, then we have the adherent cells, and I know that adherent cells are more sensitive to shear stress than suspension cells, right? So can you tell us why that is the case?
Okay, for the adherent cells are the ones that need to attach to a surface to grow. It's like different for suspension cells too, like they float around in the media, but adherent is different. Like adherent cells stay fixed on the spot. So for example, like if we use the standard bioreactor like stirring or bubbling, but to grow the adherent cells, it creates the strong shear stress, so they can damage for the cells. So and that's why the adherent cell is more sensitive to grow. Like they can just move with the flow, the media for the bioreactor, but It's like they need a calm and they need a stable for the environment, but they can grow properly, functioning, and the others.
All right. So if the sheer stress, as you've mentioned, it's really a big issue, particularly to adherent cells. If it is such a big issue, how does the industry try to deal with these problems, and how do these efforts fall short?
Well, the industry has a few ways to reduce the problem, like low flow system or the like steering more slowly or using the pedals, but they don't fix the core problem what it is. I like that.
Okay, so it's like they're trying to make an environment that is a little less harsh.
But this method still depend on moving like the nutrients or the aeration, but they don't fix the role called the problem like for the.
I understand. So it's more like a damage control than actually being a true fix.
Yes, but this method help, but they don't solve the problem. That's why alternative system like our tight motion system is actually can be helpful for different approach to help the grow cells.
All right. So with all those limitations in traditional systems, where do we go from here? What's the better path forward?
Okay, we need more than just gentle old system, but we need new way of thinking about how we grow cells, like especially the sensitive like adherent cells or stem cells. So instead of using steering bioreactor like steering tank system or bubbling system that used to bubble in the bioreactor, we use the technology like tight motion, use a gentle wave like a flow to deliver nutrients without causing damage.
Okay, so it's almost like a slow rhythmic, wave washing over the cells.
Yes.
All right. Okay, so it's not just about keeping the cells alive, it's also about helping them to thrive.
Yeah, that's right.
Yeah, okay. So you've mentioned about the tide motion principle. What's the science behind this? Can you explain why does tide motion work so well for sensitive cells?
Okay, for the tight motion is like the technology that special culture system that like they move like the natural tide. So instead of using steering or using bubbling, they gently move to liquid up and down over the cells, just like wave going back and forth.
I see. They're like in a more stable, calm environment.
Yeah, that exactly yes, but that gentle wave like motion to make the low shear environment, which is it's really perfect for sensitive cells like stem cells or adherent cells to make the vaccine or anything else that we can the surface to grow the cells. So it still deliver nutrient for the oxygen well, and so that can damage for the cells.
All right. So with all this, in a way, can you say that it's more biological than mechanical?
Yeah.
That's the goal. The system is built to match what cells actually need, not just fit the process.
All right. So how does the tidal motion actually help with, say, things like nutrient delivery and waste removal? Because we have what we call, in our previous episode, a linear scalability. So in relation to linear scalability, how does the tidal motion actually help with things like nutrient delivery and waste removal, especially when you scale up?
Okay, for the scale ups, like tight motion is like up and down to spread the nutrients and oxygen evenly across the cells. So like the best part is like they work the same way if they scale up. So they then use impellers or bubbles to create turbulence. So the cells stay in low stress, stable environment, which is it's really important for the adherent cells or stem cells.
Okay, so from what I am getting with all this sheer stress and what they do, how they damage the cells, when we have an environment such as the tide motion system, it lowers the shear stress and it could offer a reduced cell damage. So when we're talking about reduced cell damage, what does that actually mean for the final product? Like how does it affect batch quality and reliability?
Okay, for that it's like it's mean we get the more healthy usable cell at the end, and which is to leads the better and more consistent batch quality. So it's like I think it's more consistent really matter for the cells grow.
Okay, so it gets more, reliable. For the when we're talking about reliability, for the patient's life, for instance, reliability is very essential. So yeah, all right. So let's talk about what happens inside the cells. Like, how does reducing stress help improve the overall quality of the cells?
When cells don't have to fight off the shear stress, they can use their energy to growing, functioning, or useful for the protein. So that leads to healthier cells with a more stable genetic. Like, is it important for the therapies where consistency and performance matter? So like it's simply, but the better result.
But there's less stress, there's better results. Okay, so really, I think that it's more on not just about growing cells, but growing better cells. Is that right?
Exactly, with the tight motion, it's like we're not just growing their faster, but we also improving their quality. Like the cells is more come out uniform, like more functional and meet clinical the grade for the standard, and we can do all that in less time without sacrificing quality. So it's like mix of speed and reliability, which is I think it's really important for the mixed grow cells.
All right, especially for therapies where timing and consistency really matter. Okay, so what kind of real world results have we seen? Like, for instance, like, are there any observable benefits compared to, say, traditional stirred tank systems?
Okay, for the tight motion bioreactor, we have built a scalable platform using PET macrocarrier to expand MSC from various tissue sources. So like we keep the optimized seeding, the media, and they can parameter like glucose and pH. So ensuring consistent quality like for clinical and commercial use. So like it's different from traditional stir tank, like we use that stirred like so often exposed cells. If we use the stirring is like to make the high stir stress.
All right, so tide motion helps avoid all those. Okay, all right. So would you say that with this system, with the tide motion system, we're actually unlocking the full biological potential of these cells?
Oh, okay. For the tight motion, it's like when we give cell the right environment, low shear stress, and steady access to nutrient and gases, they can work the way they're meant to. Like different for the traditional system, like if we use the steering, like they are a lot of their energy to dealing with the stress. So if we use the tight motion, is the stress is taken away, so we don't worry about the shear stress.
Okay, so the full potential is really unlocked. Okay, so that's actually really fascinating. Very, very fascinating. Now, can you walk us through how exactly the tide motion bioreactor works? Like, say, for example, by using the Tide XL model that we have here in Esco as an example, how does or what does the process actually look like?
Okay, for the example, it's like Excel is like they principle they follow is like for the tight motion technology, like they up and down for the culture medium. So first, like about the nutrition is like when the metric vessel is filled with the culture media, so they are exposed to nutrition. So and in the other hand, if the culture medium is pumped back to mixing tank, the cells are exposed to aeration. So the oxygen and carbon dioxide is exchange occurs. So the cells alternately exposed to nutrition and aeration, so providing the optimal grow well for the cells.
That's actually very fascinating. So it's really about precision and creating the right, rhythm for the cells. That's what I'm getting here.
Yes. So they like we're we are going to like give the cells a calm, optimized for environment, and when we do that, they perform at their best for the growth for the cells, like especially as like sensitive one, like adherent cells or stem cells or anything else that they sensitive for.
All right. So Fitra, all the things that we have talked about in today's episode, they're very much insightful. So thank you very much for all your inputs. Thank you for joining us in this episode. All right, so now we have seen how the tide motion system of Esco could actually help your most sensitive therapeutic cells thrive and not just survive. If you want to know more about these next generation bioreactors, visit www.escolifesciences.com. Once again, this is Sheena and this has been Inside the Sciences. Don't forget to subscribe. Bye!